during the 2 years that you're doing your required practical, you will move through several stages. Firstly, you're going to need a lot of help, and the teacher will give you all the information that you need, and as time progresses, they'll start taking that help away. So at first, your teacher might even show you the experiment, then you're going to copy it. Then you'll move on to practice with support. So I do it by myself, but I need to ask my teacher every now and again. And if it goes wrong when I'm stuck, you'll ask your teacher. Then you'll be able to conduct the experiments without any support. And then basically during the final phase, what's going to happen is you're going to be able to research different methods, come up with your own, conduct the experiment by yourself, evaluate your methods, and you'll need very, very little teacher support. CPAC stands for Common Practical Assessment Criteria, and they are the same across all exam boards. Now, some of them will have chosen different practicals to be able to demonstrate the assessed criteria, and we also have sub-criteria, so that means that they're split into A, B, C, D, whatever. So for the first CPAC, of which there are 5 in biology, we have to be able to demonstrate that you have correctly followed instructions to carry out the experimental technique or procedures. Now you need to be able to consistently do this. So what we're saying is, as the time moves on through the 3 years, can you do it basically? So we get a feel of that as a teacher, and it's the kind of thing that we're marking you in class when we watch and observe you do the experiment. For the second one, applies investigative approaches and methods when using instruments and equipment. So specifically, what would you be expected to do? Well, correctly use appropriate instrumentation, apparatus and materials to carry out investigative activities. So experimental techniques and procedures with minimal assistance and prompting. Okay, so this includes ICT, so manipulating your results perhaps, or using colour emitters, or some other. Okay, carries out techniques or procedures methodically, in sequence and in combination, identifying practical issues and making adjustments when necessary. Okay, so if you can see that something isn't quite working, if you see the temperature's changing and you're not monitoring the temperature, or that the time, I don't know, thinking about required practical 2, when you had to leave the cells in acid for a certain amount of time, if you found that that wasn't working for you and you adjusted it, you increased the time or you decreased it, that's an example of that. Identifies and controls significant quantitative variables where applicable, and plans approaches to take account of variables that cannot be readily controlled. So some things can be examples of both CPAC criteria. Select appropriate equipment and measurement strategies in order to ensure suitably accurate results. So you might, for example, need to measure the temperature using a thermometer that has a resolution of half a degree instead of a whole degree. Identifies hazards and risks associated with these hazards, making safety adjustments as necessary when carrying out experimental techniques and procedures in the lab or field. As you look through some of my other videos where we look at students' work and where they have circled where they feel they have met certain CPAC criteria, you'll see more examples of this. Makes accurate observations relevant to the experimental or investigative procedure. So that can lead to evaluating your method, evaluating your techniques, anything that you've learned along the way, you're going to record that in your lab book. Obtains accurate, precise and sufficient data, so we're talking about repeats, for experimental investigative procedures, and records this methodically using appropriate units and conventions. So we're talking about constructing a table for your results, using a solidus to separate the title of the column from the units used, making sure we use the units appropriately and the correct units. Uses appropriate software and tools to process data, carry out research and report findings. So any ICT things that you use, perhaps bar charts, perhaps histograms, perhaps just manipulating your data through software. Sources of information are cited, demonstrating that research has taken place, supporting planning and conclusions. The more you're able to embed extra research in your work and you actually date and cite that work, then that's going to help to ensure that you are awarded the CPAC for that criteria. This is called an endorsement tracker, and it's given to us by the exam board. And basically we've got the 12 practicals labelled there, and down the side you would have student names, and we're recording the date in there that each of these practicals took place. We need to provide that to the exam board should they ask, and of course it needs to match your lab book. I can then flick through the tracker by competency, which they're split up into sub-criteria. So for instance, this one is CPAC 5A, CPAC 5B. And I'm able to then check all my students have had ample opportunity to have a go at this. And so it's split up into the 12 practicals and then maybe some other teacher-led practicals that we've embedded to develop those skills. We record them by colour. Black just means that there is no opportunity in practical 1 to assess that CPAC. The next page on my spreadsheet shows me student by student for the whole of the 12 required practicals.